Hi, everybody. Welcome to Chapter 4 in our microbiology class. So in this chapter, we're going to talk about nutritional requirements that our bacteria need to grow, and then we're going to also talk about how they grow, so how they replicate and what they look like, what their growth looks like in a laboratory as well as outside of a laboratory. So remember from chapter 1, we talked about Robert Koch, and he's the individual who determined that there were bacteria that caused specific diseases, and you could identify the disease-causing bacteria using his postulates, and we still use those postulates today. In doing this, he was able to develop, or he had to be able to develop methods to grow colonies of bacteria from single cells. And so initially he tried using potatoes, but potatoes aren't very nutrient rich, and so many bacteria couldn't grow on them. He then used the nutrient broth that he had, and he tried to solidify it using a gelatin, and this helped, but gelatin melts at not a very high temperature. And many bacteria can digest the materials in the gelatin, turning it back into the liquid, and you can't form colonies in a liquid. It wasn't until 1882 that Fanny Hess, which I talked about her as well, suggested using agar, which is what she used in her jelly. And this is how we now grow our bacteria forming colonies, is on nutrient agar. So like we use nutrient agar, tryptic soy agar, and then there's a lot of other agars that we use, which I'll talk about. So we can find microbes everywhere on Earth. Some microbes grow in very extreme environments. So places like volcanoes, hydrothermal vents. Many microbes, though, grow in the same places that humans and other animals live. Each species grows under certain environmental conditions. So they grow, but they are limited in growth by the amount of nutrients that are in the environment. And so if we know what the microbes need, then we can grow them in a lab, and that's really important so that we can then utilize them in medical microbiology, in nutrition, in industrial microbiology as well. So how do microbes grow? First of all, microbes in general grow via or divide via binary fission. So you've learned about mitosis in general biology, where cells divide. Well, binary fission is a similar process in which prokaryotic cells divide. They divide exponentially. So if you start with one cell, then that cell is going to replicate its DNA. It's going to elongate, and then it's going to divide into two identical cells. The time that it takes to replicate one cell into two identical daughter cells is known as generation time, and this is going to vary vastly among different organisms, and it will be dependent on the environmental conditions. This also is going to determine how fast an infection can actually occur, or a disease can actually start occurring. So we can calculate the growth of a given microbe using this equation: n sub t equals n sub 0 times 2 to the nth. The n sub t is the number of cells in a population at some time. N sub 0 is the initial number of cells that were there. And the nth is the number of generations. that have occurred. So let's look at an example. We have some pathogen in potato salad. This potato salad is at a picnic and it's been sitting in the sun for 4 hours. So we have 10 cells that have a 20-minute generation time. So n sub 0 would be the number of cells in the or is the initial number of cells. That's the number of cells we have. So that's the n 10. The nth is 12, and we get this by taking, we have a 20-minute generation time. So there's 3 20-minute periods in 1 hour and 60 minutes. So we have 3 divisions per hour, and there's 4 hours. So that's 3 times 4 is 12 divisions total. So this is our new equation. N sub t equals n sub 0 times 2 to the nth, or n sub t equals 10 times 2 to the 12th. So n sub t equals 10 times 4,096, which means if we multiply that together, n sub t is 40,960 cells of some pathogen in 4 hours that are in that potato salad. So 10 cells probably won't cause a lot of problems if they're distributed out. Someone might get a bacterial pathogen, they might not. But when you have 40,000, you're more likely to get those pathogens in your system. And so this is just showing the time in minutes. So 4 hours here. This is showing the initial population. That doesn't change because that population didn't change. This is showing the generation time, and this is showing how to, This is showing how to figure out that generation time, or the, not generation time, I'm sorry, the number of generations. And so then this is the number of cells in the population, okay? So now I'll let you have some fun, and you can try and figure this one out on your own. You're cooking chicken for a party. You chop the chicken up on your plastic cutting board using your favorite kitchen knife. Your cutting board is moved to the sink where it sits for 6 hours before you wash it. So on the cutting board are salmonella bacteria that came off of the raw chicken. The number of bacteria on the cutting board is 2.6 million cells. How many cells were in the original population? And let's assume salmonella has a generation time of 40 minutes. Now, I would pause the video here and try and figure this out, and then I'll go over the answer, okay? All right, let's look at the answer. So remember, you're using this equation here, and here what each of those items mean, or those n's mean, okay? So we started with 2.6 million cells. We don't know n sub 0. We know the end result. We only know, that's what we know. And we know that there was 9 divisions. 6 hours equals 360 minutes. So if we take 40, which is the generation time, 360 divided by 40 equals 9. So we have 2.6 million equals n sub 0 times 2 to the 9th power. So that gives us 2.6 million equals n sub 0 times 512. If we divide both sides by 512, we started with 5,078 cells in the initial population. I'm going to stop here, and we'll get into growth of microbes, okay, in the next video. Bye.